remnants of bacterial DNA that has colonized the bone after death, at some point after death. So in our studies, our main interest is in the human fraction, and if there is 1% or more, so most of the time, for most samples, finding even 1% of preserved DNA for Neolithic Anatolian samples, that's already pretty lucky. For many samples, I could roughly say perhaps 40–50%, we don't find anything at all, any human DNA, practically no human DNA preserved in those samples. So then we have to, we cannot do anything with those, with that material anymore. No genetic information available. For those samples where there is human DNA, some indication of human DNA preserved, there's additional steps to study authenticity. I won't go into detail, but I could try to explain more if you wish. So there are a couple of ways to do that, but once we are confident that the DNA, human DNA, is authentic, is indeed ancient, and if there is, well, 1% or close to 1% of DNA left, then we continue producing more sequences, and that eventually yields enough data to be analyzed for population genetics related questions or kinship related questions. Yeah, there's here an alternative way that some labs prefer capturing DNA, so enriching the libraries for human DNA. That's something we usually don't do. But in the end, in either way, you end up with basically a data set of individual genotypes, so the ATGC of DNA, basically, which you convert into ones and zeros eventually, with a lot of missing data, because for most samples, it will be prohibitively expensive to produce genetic information from across the whole genome, but usually 2% or 5% of DNA just distributed randomly across the genome is enough to answer many of the questions we're interested in. So that leaves us with many individuals represented by only partial genomes, although in some cases where in samples where there is high amounts of DNA preservation, and although it's super rare, we have come across some Neolithic samples that have 40, 50, even 60% human DNA. So 60% of the DNA molecules we sequence belonging to human. This sometimes happens from those rich samples. We can also we can produce more data and produce more high quality genomes. Of course, an important point here is we, in all our analyses, we don't just use the data we produce, but we combine it with published data. So there's this nice culture in the field that people, when publishing their papers, they also publish the raw data, or at least some form of raw data. And so one can combine one's own data with what's published and continue. So within this project, again focused on Neolithic Anatolia, we've sequenced about 800 skeletons for now across 32 sites, and about 200 still remain to be sequenced, and that has yielded us already about 200 usable genomes, and about half of those actually are from Çatalhöyük. But most of the analyses are still ongoing, so I'll be able to talk about only a small part of this data we've produced. Most of our data till now is coming from Central Anatolia. So our richest collections are from Central Anatolia. We're also studying the West, and also, of course, very exciting topic is the Neolithic of North Mesopotamia, so Southeast Anatolia and Zagros region. But DNA preservation in this area is much worse than Central Anatolia and also the West, in fact. So we have currently the most of the data available is from Central Anatolia, and so the story I'll be telling you will be focused on in this region. And as you probably all know, I mean, Central Anatolia is actually not in every sense a part of the Fertile Crescent, but it was a region that started where we see signs of sedentary life, villages, and some plant cultivation, including domestic plants and animals, pretty early in the process of Neolithic development in Southwest Asia. So sites like Aşıklı and Boncuklu, they were already established within the 9th millennium BC. So the story starts not as early as in the South Levant and North Mesopotamia, perhaps, but it's also pretty old, much earlier than the story we see in West Anatolia. And I'll be talking about data from these 5 different sites. We've produced data from many of them, except for Pınarbaşı, and some data from Boncuklu has been published by other groups, which we've been also analyzing. Just to again introduce you the sites. So the Pınarbaşı genome we're talking about, it dates back to about 13,000 BC. It's pre-Neolithic. Pınarbaşı also has other phases, but the genome belongs to the Epipaleolithic phase. From the Aceramic or PPN Neolithic, pre-Pottery Neolithic, we have Aşıklı Höyük and Boncuklu from two parts of Central Anatolia, Konya, the Konya Plain, and Cappadocia. And our genomes date within this time frame, about 8500 to 7500 BC. And from the ceramic pottery Neolithic period, we have Çatalhöyük and Çiftlik genomes studied, and they fall in a time frame between 7000 and 6000 BC. And by this period, as most of you know better than me, we have full farming communities in Central Anatolia. So I'll also be referring to Çayönü as the only published, for now, well, not, I shouldn't say published, it's a preprint, but the only report of which is available from the North Mesopotamian region. So this is a plot from that preprint where we were presenting Çayönü by Ezgi Altınışık and others. And I'll just try to introduce you to the different colors and the points. So this is a multidimensional scaling plot of genetic, it's a summary of genetic distances in 2 dimensions. Closer the points, the more similar they are in their genetics. And here we have Natufian genomes and PPN Levant genomes from Ayn Ghazal and other sites. And they cluster together, as you see. So here we have Central Zagros Neolithic genomes and Caucasus genomes. So Caucasus and Central Zagros, they are genetically, South Caucasus from the early Holocene, and Zagros genomes from, again, the early Holocene. They are genetically very similar to each other. So we and others frequently treat them as a single gene pool, as a single population. And here we have Central Anatolians in true cause. So different groups. this is the Pınarbaşı genome. Perhaps I have here. These are Çatalhöyük and Çiftlik genomes plus Barcın, but which genetically is pretty similar to the Central Anatolians. And here we have Aşıklı and Boncuklu. So in time, we see a distinction over time, as you see. So from Pınarbaşı to pre-pottery Neolithic Central Anatolia, there seems to be some amount of shift. And in when we come to the pottery Neolithic, we again see some kind of differentiation. At least the plot implies such genetic changes. And finally, Çayırını here is located somewhere in the middle between Anatolia and Levant, but also is slightly shifted towards the Zagros. And this shift, we'll be talking more about that, but it implies some amount of admixture, more eastern admixture in the Çayırını genomes. Okay, so here I'll be presenting you some very basic observations we and actually others have also made based on this genomic information. So for one thing, in the very early Holocene, or even the very late Paleolithic period, the genomes that have been sampled, that include Balkans genomes, the Natufians, and also Caucasus and Zagros genomes, we see pretty strong differentiation, except for the similarity between South Caucasus and Zagros. So these groups appear pretty much differentiated from each other. One point here, though, to make is that the Pınarbaşı genome actually can be explained as a mixture between the European, the Balkan, early Holocene or pre-Holocene groups, genomes, and the Levant groups. So both our analyses and others, well, the data was originally published by Feldman et al. Their predictions and also our analyses suggest that there was some time before the Holocene, there was some admixture events that might have happened in or around Anatolia that involved the mixture of Western and Eastern groups, Eastern here represented by the Natufian, that led to the emergence of an Anatolian gene pool. It's unfortunate that this period is represented by only a single genome in that period, but multiple analyses pretty convincingly suggest that this, what the Pınarbaşı genome is suggesting, this admixture event, perhaps that might have happened between 20,000 to 50,000 years ago, that it's real. It's factual, but we should, of course, still we have to be cautious in our interpretations because our representatives here from this region are also relatively small samples. But yeah, most likely what Pınarbaşı represents was a representative of the Anatolian very early Holocene gene pool. With, of course, more data, more samples, we'll know better. And when we compare Pınarbaşı with later, so the early Neolithic, Aceramic Neolithic groups in Central Anatolia, Aşıklı and Boncuklu, we find more or less, we find largely genetic continuity, but some degree of Eastern gene flow. And perhaps I have, let me use this graph to try to explain what the pie and what the colors actually represent. So imagine there's one population, one internally homogeneous population ancestral populations. Here, time is flowing from top to bottom. That for some reason splits into two sometime in the past. And the two groups, the two daughter groups, they remain isolated for some time. Here I just randomly, arbitrarily denoted these west and east. So if these groups remain separated for enough time, then through a process called genetic drift, which is basically named for random events, you eventually end up with genetic differentiation. Differentiation between the two groups, even without, we don't have to assume any adaptation, any selection, anything functional. Just as long as these groups remain isolated for long enough time, they will become genetically different. The smaller the groups are, the faster this differentiation process will happen. The larger they are, the slower it will happen, but eventually it will happen. And that's what the colors here are representing. We have they both come from the same original source, but over time, through random events, they have differentiated. And then imagine an event of, while these remain mainly isolated, a migrant group, for whatever reason, from the Western population goes and joins the Eastern population, and they mix, so they mate, they have kids, and that will introduce these Western alleles or Western type genetic profile into the East, and that will be already observable by the first generation. Of course, you know, how such migration events might actually happen. I mean, it doesn't have to, for one thing, it doesn't have to happen in one bout. It doesn't have to be within a short time frame. Irrelevant to how exactly such human movement happens, if about a quarter of the ancestors of this group actually come from the West, we will observe it in their children's genetic profiles. And that signature of, while they will continue, of course, if they remain isolated, while they continue differentiating, that admixture signature actually remains in the gene pool. So it's still detectable in their offspring and grand offspring. So that's what these colors are telling us. The green represents the Pınarbaşı genome, the early Holocene or pre-Holocene Anatolian groups, and the purple represents Eastern groups, here only represented by South Caucasus and Zagros. So where exactly this Eastern contribution happened, we don't exactly know, but the given data suggests that most likely this was some kind of Eastern gene flow. And we can also, a small parenthesis, look at Çayönü. This is again data that's reported in a preprint. Çayönü is quite alike the Anatolian Aceramic populations, but it looks a bit more Eastern, and also seems to contain a bit more this light green representing Levant genetic profile. So it's more admixed than the Central Anatolians, which is consistent with its more central location. Here it's, sorry, the pie is shifted a bit up, but I guess you know. So yeah, what the data is telling us that Central Anatolia, within this period, it most likely received Eastern gene flow. The exact source and when this happened, how exactly it happened, we don't know. It might coincide with the transition to Semitism. We would only be able to answer that if we have more data. There are ways to estimate the date of admixture, but they don't always work so efficiently. And one thing which is, I believe is particularly worth noting is that, so Aşıklı Höyük and Boncuklu, they were both Aceramic Neolithic, but they've the archaeological excavation teams have described many differences in their cultures, from architecture to symbolism, animal management, for instance, very well documented from Aşıklı, not really known from Boncuklu. So one thing that is clear is that both Aşıklı and Boncuklu, they were genetically very similar populations, so their demographic history was very similar. And so there is no obvious difference in their demographic histories that seems to correlate with their material culture differences. That was one question we had in this project, but I'll be showing you more examples. But overall, material culture differences among contemporary settlement sites, they seem not to be really correlated with differences in demographic history of these populations, at least large-scale differences. Yeah, so now we come to the ceramic period. We have two representatives for now. We also have Musular, that's not shown here, which is like on the border between Aceramic and ceramic. And we have Çatalhöyük and Barcın. Here, just for simplicity, I've used the same proportions, some kind of average proportion. Don't take them at the face value. They're just to give you an idea about what we're seeing in the genetic data. So what we observe is, again, large degree of continuity from the Aceramic to the ceramic period. But again, we observe evidence for gene flow, mainly from the east, or both the east and south, or a simpler solution, actually a simpler way to explain data is inferring admixture from North Mesopotamia, here represented by Çayırını. And indeed, if we study ceramic Neolithic or proto-Neolithic populations in Anatolia and check their Çayırını affinity versus South Caucasus affinity, Çayırını appears as a much much better source, better fitting source to explain the observed genetic change than South Caucasus, for instance. So here we have some indication of the potential source of this gene flow. It corresponds to the transition to full farming, whether this was related, this human movement was related to the change in material culture and lifestyle. We don't actually know. Whether it was related to the so-called PPNB collapse in the Fertile Crescent, that's also something that needs to be further studied. One thing perhaps I should note out is that thanks to the Musular, Aşıklı and Musular data, so the Aşıklı genomes, we have the latest ones, they go down to 7500 BC, and the Musular genome, it's dated to about 7200 BC. So, and we already in Musular, we see this genetic change that we further observe in Çatalhöyük and Barcın. So at least a significant part of this human movement seems to have already taken place within this time period, which is interesting. It doesn't seem to have been a totally gradual change that spread from 7500 to, let's say, 6000 BC. And we again, with respect to their demographic histories, there are some differences between Çatalhöyük and Barcın, but nothing very obvious, nothing that we can directly attribute, can use to explain material culture differences. Okay, so some overall points. We observe genetic continuity in Central Anatolia to a large extent, but there are some changes. One intriguing point is that we have genomes that cover approximately 1,000 years of Aşıklı's occupation and 1,000 years of Çatalhöyük's occupation. The letter isn't published yet. And in both cases, we don't see we don't see any large-scale migration from distant sources from the east or the west. Of course, it's very possible that both groups actually received people from Central Anatolia, from regions where people with population with similar genetic profiles lived. But distant migration we don't see within with our genetic data for now, at least. But there are some mobility events that we can detect. The one that seems to happen around mid and late 8th millennium BC is probably one of the more striking ones, which might be related to North Mesopotamia. One other point that might be worth making is that the main source of human movement in both the start of the Aceramic Neolithic in Central Anatolia and also in the transition from Aceramic to Ceramic Neolithic, the main source of movement seems to be the east and the south. Although we have to, I have to mention that we don't have yet very good, at least published representatives of genomes from the Aegean period, pre-Neolithic genomes from the Aegean period, which I hope will soon change. Yeah, so another observation is that among groups that are contemporaries, both in the Aceramic period and Ceramic period, there seems to be limited amount of genetic differences, which makes sense given that cultural change, dynamics of cultural change are usually much faster than demographic dynamics in many settings. Of course, not always, but in many instances. But we also have to note that if some of these settlements were receiving gene flow movements from also distant sources, but at levels of 1 or 2% of migrants, for instance, coming in every couple of hundred years, such low frequency of migration might still be sufficient to introduce new methods, new ideologies, but it might not be visible with the type of data we're currently producing, or with the amount of data we're producing. So we cannot rule out, we don't rule out human movement, also distant human movement, of course, in periods when we say there is limited change, but the movement might just be at too low levels instead of mass migration, might be just limited amount of change. And that perhaps is that kind of idea that movement still continued but left a limited impact on gene pools is supported by our recent work on Çayönü. So you might have noticed in this graph that the Çayönü samples we produced were clustering together, but there was one outlier looking genome, infant girl, and she appears to carry much more Eastern ancestry than other Çayönü individuals, and we estimate she has about 20% extra Zagros ancestry. And we predict that she had actually relatives within the settlement. So we could predict that her paternal ancestry probably was more local, but at least part of her maternal lineage was of more eastern descent paths of North Zagros or Central Zagros. And this individual actually dates in somewhere in the middle of our temporal sample. So she is actually a representative of migrants coming in to the community. But overall, across Chayunu's occupation, we also don't see a major shift in ancestry. So obviously, our power to identify such movements in large-scale data, unless we find such, we're lucky enough to find such migrant descendants, our power is limited. Okay, so I'll also briefly now talk about, switch to this topic about kinship. So this question of whether individuals buried in the same spaces in Neolithic societies, where we find quite frequently burials within buildings or next to buildings. That's been ongoing, that's been debated for a long time. And it's an important question because it also relates to how sedentism might have started. So ethnographic work suggests that mobile, pre-Neolithic mobile hunter-gatherer groups, they might have been not organized solely based on genetic kinship. The role of genetic kinship might have been limited in group organization, but there is this theory that with the start of sedentism might have involved organization that is much more based on genetic family groups, nuclear families. In fact, and there's also the hypothesis that these evolved into extended family groups in later Neolithic periods. There are multiple. I mean, these are not. These are just hypotheses for now. But yeah, so quite a few anthropologists and archaeologists assume these could make sense. Such a model could make sense. But of course, finding evidence is not super easy for this type of question. Until recently, the main source of evidence came from Çatalhöyük, which has a huge and very well curated human remains collection. Çatalhöyük, as you know, was from Central Anatolia, stored the Ceramic Neolithic, and like many other Fertile Crescent Neolithic communities, there was a strong culture of intramural burial, both adults and subadults, and a work done about 10 years ago based on dental morphometrics and also traits, so qualitative traits, which can inform on some degree of, which can inform on relatedness, although being very noisy. It suggested that people buried in the same buildings didn't appear more close to each other, biologically more close to each other than people buried across different buildings, which was, of course, very interesting, provocative, but because it was dental data, many in the field remained a bit suspicious. So recently we went back to this question. It was also one of the main questions of the project, and both using data we produced and data others had published, we combining it with burial information in collaboration with the excavation teams. We tried to infer the level of genetic relatedness among individuals buried in the same spaces, either the same building or neighboring buildings. And I'll walk you through a couple of examples. Here we have Aşıklı Höyük, early Neolithic in Central Anatolia again. And we have two round buildings from these are from the earlier levels. And among the 5 individuals from which we could get DNA data from, 4 of them had relatives. This young adult female didn't have relatives, but the other 2 were inferred to be siblings. And here we have Boncuklu Höyük, again, 2 neighboring buildings, again, relatives, a parent, inferred parent-offspring pair, and a sibling pair. But again, not everybody has relatives in the group. This is a perinatal baby buried with this woman, and when the excavators found the grave, they assumed that the baby must be that of the woman, but it turned out that the baby was totally unrelated. We haven't found any relative of this baby yet. Okay, so this was the Aceramic period. When coming to the ceramic period, we observed something interesting. So I should note that in Barcın, so our samples were from Barcın and Çatalhöyük. In both sites, the burials we could obtain enough DNA from for doing genetic kinship analyses were mainly sub-adults. And here we see 3 buildings from Barcın, and overall we could find only 2 pairs of relatives, but quite a few individuals turned out to be unrelated, at least not related beyond the 3rd degree. And in Çatalhöyük, a similar sample size, again, 3 buildings, we could only find 1 pair of siblings. So quite a few children, infants, babies buried in the same building, but not being genetically related. We also studied more distant relationships where the individuals from the same building could be more, not perhaps 2nd or 3rd degree, but more distantly related to each other compared to those in other buildings. We again found no such indication. So overall, there seemed to be a difference between the Aceramic Neolithic sample we had and the Ceramic Neolithic sample, in that in the Aceramic Neolithic co-burial groups, the frequency of individuals where we could find at least 1 relative was higher. In Çatalhöyük and Barcın was smaller. So this was exciting for us, but of course we had to remain cautious because given the pretty small sample sizes from each of these settlements, but our more recent data seems to be supporting this initial conclusion. For one thing, in Çatalhöyük, we've again obtained data from 3 buildings. The ones shown in red, not pink, are the individuals where we could obtain enough data from. Again, there are some cases where we don't find relatives like this individual, but for the majority of individuals with usable DNA, we can identify relatives. Çatalhöyük again goes to the PPN period. And I'm not showing you this, but our data from Çatalhöyük likewise support that in later periods. There seems to be a decrease in the frequency of relatives buried in the same buildings. Of course, our data, I already mentioned that our Barcın and Çatalhöyük data, they're mainly from subadults. Seems to be an issue with DNA preservation, at least in these sites. And so what we're seeing might be also at least partly related to traditions revolving around the subadults infants. And in Çatalhöyük, for instance, we already know that they were subadults were treated differently, especially babies, infants. They were buried mainly in around the oven area, in the so-called dirty area, whereas adults were buried mainly under the cleaner platforms. Yeah, so it seems that the traditions around how individuals from the same biological family were buried, whether they were buried together with those from the same biological family or not, that might have varied in time. At least there's some kind of the traditions are seem to be not uniform, either time or space. But of course, with more data, we'll be learning more. Okay, and the last thing, I just want to introduce you to results from a recent study we submitted. Now, not just looking at the Neolithic period, but across the whole Holocene, using large data set, mainly published, but where we also included some new genomes. In total, we have about 400 to 500 genomes from this region, Southwest Asia and the Aegean. And here you see the distribution of the samples. And our question was how, well, in the Neolithic, we already discussed how human movements can be observed in different periods. We see the gene pool in Central Anatolia, for instance, receiving Eastern migrants. So how, our question was what we can infer from the whole region and across the whole time span. So can we observe some consistent patterns of human mobility as inferred from genetic data? So one pattern we observe is genetic diversity increases in all these regions. We observe that the genetic diversity is increasing over time. And this was something that people have been observing for the last 5-10 years, repeatedly in also other parts of the world. And this increase in diversity, it's best explained by admixture from outside sources, from outside the region of interest. The x-axis is time, as you might have noticed. We further though notice that this is a representation of, again, genetic distances of individuals from different regions, shown with the colors here, and in different periods. So one thing we observe is if we study genomes from more than 10,000 years ago, they're pretty distant from each other in the plots. So here we have Levant, Anatolia, Caucasus, and Iran. And then as time goes by towards, with the Neolithic transition, we see a convergence of these gene pools in the principal, this is actually principal components plots in the PCE space. And what was more interesting, perhaps, was that this convergence, by the way, it can be explained by, let's say, Anatolia receiving Eastern gene flow and Iran and Caucasus receiving Anatolian gene flow, which has been described by many groups recently using ancient DNA data. What was more interesting for us was that when we measured interregional genetic distances, so distances among these regions on average, each pair of regions, and plotted that against time, we observe, as what the previous slide implied, we observe a decrease in genetic distances until 8 to 6,000 BP. But then following 6,000 BP, so basically with the Bronze Age in Southwest Asia, we see a trend of increasing genetic diversity among gene pools. And this implies that admixture continues, but the sources changed, and now these regions receive gene flow from more distant groups. So in the Aegean, for instance, we know that after the Bronze Age, or with the late Bronze Age, there is an influx of steppe-related groups, and in Iran, in the Caucasus, similar changes have been described, not in Anatolia, but Anatolia also receives gene flow from more distant regions. So overall, there seems to be a shift in the scale of human movement and admixture with the Bronze ages. That starts with the Bronze ages and continues till the present day. So that was something we thought was interesting. And with that, I should thank all my collaborators, the whole team who produced the data and analyzed the data, and other members of our project, Çiğdem, Yilmaz, and Elif. And of course, the excavation teams who have been kind enough to share both their material and the archaeological information. And yeah, so with that, I should stop. Sorry if I took too long time.